started. My name is Dr. Megan LaFollette, and I am Executive Director at the 3Rs Collaborative. And today I'm excited to welcome you to the 2024 Microphysiological Systems webinar and workshop series. Today's workshop will be focused on MPS cell sources and suppliers. As a disclaimer, this webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subject, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. So to start off, and just in case people are unaware, I want to give a brief introduction to the 3RC. We're going to give an introduction to IQ as well as this webinar series. So the 3Rs Collaborative's Microphysiological Systems initiative is really focused on collaborating to accelerate the adoption of MPS in scientific research. On the screen, you'll see our website as well as our email address. And actually, you'll see my program manager who typically usually runs these webinars. Her name on the screen. She was unable to make it today, so I'm kind of stepping in. Our members really span across the scientific field. This is a picture of just our leadership and board of directors, as well as their institutional logos. And you can see it really spans across both industry, academia, nonprofit, government agencies, and more. And our initiative on MPS really seeks to increase adoption as well as regulatory use of MPS in coordination with animal studies. We have 42 institutions, 30 that are developers that are part of our initiative. And our initiative has kind of 5 key aims. First, to provide thought leadership and expert consensus from primarily developers as well as other stakeholders on MPS implementation. Facilitate appropriate discussion, collaboration, and sharing between commercial developers, develop external partnerships and collaborations, create direct engagement between developers and regulatory agencies, and create resources to facilitate increased engagement with commercial MPS developers. We have 3 key work streams that we accomplish these goals with. We have a regulatory group that publishes papers, hosts meetings, creates presentations, and works on collaborative regulatory related projects. We have a technology expo group that is all focused on our technology hub. And we have an end user and education group, which is the group that runs this coordination that both creates educational resources, hosts these webinars and workshops, and generally works on conference activities, etc. We engage consistently with end users and provide education. These are some of the things that we've done from 2023 to 2024. Some new things this year is that we've joined the All-Techs as a member and have been publishing in corners. We have our quarterly MPS workshops. We've had talks at the MPS World Summit, SOT, EUROX, etc. And one thing not on this slide is we've actually done an update to our brief and facts about the FDA Modernization Acts. We do have a presentation page where you can access all previous webinars and workshops. So if there's a topic you're interested in about MPS, I encourage you to check it out there. Our regulatory group has been doing great work this year as well. We've submitted a publication on perspectives as well as roles on using MPS for regulatory application. We also have a public-private partnership with the FDA CDER, are in the process of conducting a cross-platform project on using MPS for DILI applications, including developing a platform-agnostic protocol, experimental design, etc. We have our MPS Technology Hub that was launched in 2021. We're continuing to adding more companies. We've added disease areas and filters, and we're working on a publication hub in the future. This is what our hub looks like. You can view it on our website. It's pretty easy to access, and it's a great way to find providers that will work best for you. Our upcoming workshop after this one is going to be on November 12th from 10 to noon Eastern time, and it's going to be on oncology MPS. So stay tuned for a future invite to that. As a note, just for housekeeping, please submit any questions using the Q&A if possible, and we'll answer the questions either at the end of each individual webinar or in the roundtable. So please do direct them to specific presenters when possible. You can feel free to visit our website for more information and email our program manager, Lauren Young, if you're interested in joining our initiative or becoming one of our sponsors. And with that, I'll go ahead and turn it over to David from the IQ.
Thanks, Megan. I'll go ahead and share.
Great, we can see and hear you, and we see your slides. Go ahead.
Great, awesome. So hi, everyone. My name is David Kukla, and I'm part of the IQ-MPS affiliate, specifically serving on the strategic partnerships and communications team. So I just wanted to kind of give a quick overview before we start on the IQ-MPS if you're unfamiliar. So the IQ-MPS is currently composed of 27 member companies, and we have representatives from drug safety, 3Rs, ADME, as well as PDPK. And currently our 2024 leadership team is made up of Anya Kopek, Rhiannon David, Kim Homan, and Rhiannon Hardwick. And besides this leadership team, we have a steering committee, and this is made up of 2 individuals from every one of our member companies. And then to the right, just highlighting a little bit more about the IQ-MPS, we have over 150 active participants, which meet several times throughout the year. We also have several publications and working on several more, as well as we have a relatively new LinkedIn page that we often post any news out of the IQ-MPS. So feel free to follow us there just to keep up. And there are 4 main goals of the IQ-MPS. One is to serve as a thought leader for both MPS developers and stakeholder organizations. Another is to provide a venue for appropriate cross-pharma collaboration and data sharing to facilitate industry implementation and qualification of MPS models. Another is to create focused engagement between industry and regulatory agencies on the current status and evolving field of MPS in an industry setting. And finally, to develop external partnerships and collaborations to help enhance the inclusion of industry priorities. So besides that, the IQ-MPS is made up of 5 affiliate work streams. There is one on organotypic manuscripts team, where the goal is to publish manuscripts outlining organ-specific industry requirements for MPS platforms. Another is our regulatory outreach work stream, which identifies opportunities to engage with health authorities globally on topics related to MPS qualification. We also have a pilot project team, and this team initiates near-term proof of concept study for data sharing or prospective collaboration for qualifying MPS systems. Another, which I'm a part of, is the strategic partnerships and communication team, which we like to build relationships with key MPS stakeholders. And then finally, a landscape gap survey team, which conducts and analyzes surveys, benchmarking how pharmaceutical companies are currently using MPS. So here, I just have an overview of our external partnerships team over the past few years, starting back in 2018. However, as I mentioned, we do have several other activities within cross-pharma collaboration and data sharing, regulatory agency engagement, as well as thought leadership. But some of the highlights from our external partnerships team in 2021, as we already mentioned, we co-partnered with the 3Rs Collaborative and established an MOU, which has allowed us to create workshops and webinars such as this. And in 2022 and 2023, with the 3Rs Collaborative, we've had 4 webinars each year on different MPS systems, such as neuro, liver, blood-brain barrier, vascular, and GI, to name a few. And then within this year, in addition to this current webinar and 2 previous webinars on immune models, as well as neuromuscular and fibrosis models, we do have one more model that Megan already, one more webinar that Megan already mentioned on oncology models coming in November that we're excited for. So additionally to that, we have participated in different IQ-MPS FDA workshops, as well as having an upcoming webinar across organ systems with the FDA. So we're really excited to continue these efforts throughout this year and upcoming years. But if you have any questions specifically about the IQ-MPS, please feel free to reach out to us, or you could check out our website that I have highlighted at the lower left of this slide. But yeah, that's everything from me. Yeah, thank you.
So thanks for inviting me for this presentation. So I'm going to try to give you a bit of an overview how sources and supplies we use for our human multi-organ chips. So since I have the first talk, I thought I start with a bit of introduction with the cell sourcing at a glance. So usually we have adult donors where we can have primary cells, tissue biopsies, or explants from. These can go immediately or directly into the chips. We also have the possibility to use immortalized human cell lines like HeLa cells, Hyper-IgG cells, and CACO2 cells. Everyone, I think, is aware that these cells have high limitations if they are compared to the primary tissue. Nevertheless, they also have the advantage of having a very high number of highly reproducible results from the same cell. Then we have adult organ-specific stem cells where you can get organ-specific organoids from fetal stem cells, where we have them already differentiated into ectoderm, mesoderm, and endoderm-derived tissues. And of course, embryonal stem cells, which can differentiate in any cell type or organoid and have unlimited source of cells. And then, of course, since nearly 20 years now, we have induced pluripotent stem cells, which can differentiate into any cell type or organoid with nearly unlimited availability. Or unlimited, not availability necessarily, but we have the same cell source for a long time. At TissUse, we have worked with IPS cells, cell lines, primary cells, biopsies, precision cuts, tissue slices. We have developed a lot of different organ models out of these. Most of our organ models we have from different cell sources, and I'll give you an example in a couple of minutes. So just to give you a bit of an overview, what I'm actually talking about when I talk about our chips, this is our platform. It has a microscopic glass slide at the bottom. And we have a microfluidic that is a closed system, so we don't have an inflow and an outflow. Do not dilute any growth factors or cytokines that are produced. We have a culture compartment here and a culture compartment here, which are connected by a fluid flow, and an on-chip pump, which is separated by a membrane and has valves here, and works a little bit like a heart that is filled up with medium and then pumping the medium in a positive flow. One chip has two replicates in terms of the chip 2. It's suitable for iPS-derived cells, primary cells, 3D tissue, cell lines, and so on. As I mentioned before, you can see that the positive flow is depending on which area we are looking at. We've also developed it in a way to have a plug-in option for insert barrier models. This is also important if we decide to have a barrier model. We might need different cell sources. So here we've decided to work on with standard cell culture insert, but we also developed our own cell culture insert over time. And of course, the whole thing is compatible with live tissue imaging because it's microscopable. We've developed different microfluidic designs because, again, I come to this in a second. If I downscale an organ model, it's also necessary to downscale it at a ratio. So I might have a liver model that is small and a pancreas model that is also small. But for an intestinal model or a lung model, I might need a bigger surface area to downscale at the same ratio for the functional units. So that's why we have different sizes here for the chip 2. And then we have chip 2 connecting 2 organ models, chip 3 up to 3 organ models in different sizes. And then the chip 4 is a bit more complex, having a proximal tubulus part here and a glomerulus part here, and a PBPK compliant ATME chip to also have excretion and absorption in the system. This is how it looks like in the lab. It's compressed air supply with a starter and power supply, and then we have tubes going into an incubator connecting the chips in a standard incubator. And then how do we prepare for an assay? So we use the different cells, like, again, it could be any model. It can also be a commercial model. And then we prepare these models, and we have to decide on what kind of readout do we want to have at the end, like the context of use, to decide which model of these on the left side we are actually using. Then culturing for single or repeated dose for up to 4 weeks, or maybe even sometimes longer, depending on our context of use. In-process controls are possible. We can sample 250 to 500 microliters to look at metabolism, viability, metabolite analysis, and so on. And of course, all the time, also non-invasively, we can look at cell morphology and molecular imaging. At the end of the culture, we can remove the tissue. And again, here it's important what kind of tissue do we decide on working on, so that we can also adapt the endpoint to it. So may it be histology, qPCR, metabolite analysis, single cell sequencing, and so on. We might need another amount of cells for each of these than for another readout. So I've made a list, and I know that this looks like very crowded slide. But if we consider the level of biological models, it's always necessary to look at the intended context of use. So for the level of emulation of human pathology and background biology, it needs to fit to the intended use, like may it be vascularized models, innervated organ models, or immune competent systems. Then we need to decide on different cell sources. Like I mentioned before, relevant organ downscaling and physiological portions are crucial. So we need to take the tissue to fluid ratio into account, and the tissue to tissue ratios. And of course, especially because we have different organ models that we connect, we need to make sure that our model-specific culture medium is also fitting to our co-culture medium. So we need to optimize the medium according to our cells, and of course the cell source selection. If we look at the right side, for the assay level that we need to consider, we need to look at the throughput capacity. For example, for our thyroid model, we have limited sources of cells available, and then these cell sources are very limited per donor. So do we need to do an experiment on the same donor for many, or do we have a high donor-to-donor variability for our experiments? Then we have very limited access for these tissues, so we might need to go to a different cell source. And then move only after some experience to the primary cells, where we can really see a donor-to-donor variability and have different donors, but very limited amount of cells per donor. Then, of course, for our decision on what kind of cells we're using, it's the administration route is important, the dosing scheme. So do we need for the intestine, do we want to do an organoid culture, or do we need to look at absorption? Then we need a barrier model. This might actually lead to different cell source than for organoid culture. And then, of course, also another very important point is the primary endpoints that should ideally match the ones of clinical trials, like survival and tumor size and so on. At TissUse, we have different established genomic assays, and we've co-culture assays, and we've grouped them in different level of readiness, meaning our level of readiness being how far is the model or an assay established in-house, and has it been tested also in other labs, and is the reproducibility and the intra-laboratory reproducibility comparable. Oh, hi. So one of the examples where I want to give you a bit more detail about what kind of tissues we have tried is the liver model. So we've connected the liver with quite a lot of different organ models, which means also we have different context of uses here. Those who know us for a bit longer, they also know that we've worked a lot with HepRIG-based liver spheroids. This doesn't mean that we are not very well aware that HepRIG cells are a cell line, and therefore brings a lot of disadvantages, or like low comparability to the primary tissue. Nevertheless, we've used it a lot to establish our organ models or our chip models at the beginning, trying to figure out how we can actually develop the chip first and the co-culture with the skin, intestine, pancreatic islet, thyroid follicles, lung and testes, and so on, and then moved on to a primary model. So here we used InSphero 3D inside human liver microscope microtissues, for example, as a commercial model, which is composed of primary human hepatocytes from either multi-donor or single donor. Again, here it depends on the context of use. What are you looking at? Would you like to have a specific SIP production or SIP activity, or is it better to have an overview over several donors? We can co-culture, or they offer the co-culture of Kupffer cells or liver endothelial cells that is possible, which again makes a huge difference for our assays. Then we have primary human red and liver spheroids that we have also in our chips, where we use our own or our customer protocols. And I'll give you an example of the liver ring trial in a second, which we connected with pancreatic islets, cardiac spheroids, and thyroid follicles, or IPS-derived liver spheroids. And here you can see that the amount of cells that we use in the chip can vary. And this again, if you need more readouts, can vary, and you need to apply different readouts for the different tissues. So for the liver ring trial, we've did a cross-company pre-qualification trial for liver MPS models. We want to address the lack of confidence for regulatory submission and tackle reproducibility and robustness. This is in cooperation with UCB Biopharma. And here we use the liver chip, or the chip for only with the liver. And we use these little microcavities. And we actually could see that we used already cells that were noted as these form spheroids, but actually in our microcavities, not all of these cells that were noted as these form spheroid or spheroid compatible also worked in the making these spheroids either in plates or in the chip directly. So it also needs to be evaluated again in our lab, which is usually quite time consuming and quite expensive as well. Starting with a liver ring trial, Benoit and Rainer from UCB started off comparing static cultures of liver spheroid with our MPS platform, seeing that liver chip spheroids are more reproducible and have stable LDL release, indicating better viability compared to similar setup in a plate, and also stable biomarker expression in the liver chip. So you can see that the cells actually behave different in the chip than they behave in static culture. Another example that I quickly want to go over is the bone marrow on a chip, where we can actually see that an allogenic chip can be different to an autologous chip. So we had a ceramic that was cultured with MSCs and HSCs. And then depending on which kind of medium we add, we can induce different lineage differentiation. So the medium really plays a role here. We can either differentiate in a myeloid into NK cells, so lymphoid lineage, or erythroid lineage, or in a fourth medium, we can actually differentiate into all three different lineages. And these are the cells that we are actually able to measure over the time. And then in a project with Roche together, we wanted to also add T cells, because we can't differentiate T cells on our own directly from our bone marrow. So we added 500,000 pan T cells to the system and started a treatment with a bispecific antibody or T cell bispecific antibody. And what we could see is, I don't want to go into details of the bispecific antibody, but what we see is adding these T cells, which are allogeneic, reduced immediately our erythroid population. So without any treatment, we only had like 40% of these erythroid lineage in our system anymore. So we decided to try it out within, we also saw that the activation markers were already used, and then looked at a healthy donor where we had IPS cells, differentiating them into hematopoietic progenitor cells, into MSC cells. And then from the same donor, we used blood to isolate T lymphocytes into the chip. And we could see here that, first of all, on the bottom, you can see that the activation markers in the allogenic chip were much higher than in the autologous chip. And then if we also looked at the erythroid lineage cell counts, we can see that this depletion of erythroid population with the autologous chip was much lower than in the allogenic chip. So here again, the medium, the cell source, the donor is very important. And with this, I filled up all my minutes, so thanks a lot. And I'm happy to take any questions.
So hello, everyone. I am Carolina Lucchesi. I am a cell biologist, and I worked when developing organ-on-chip models for about 12 years between the Visa Institute and Emulate. And about 2 years ago, I joined ATCC to establish an MPS program, which I will elaborate in the next few slides. So in today's presentations, I'll be talking about ATCC's mission and our future directions. Quickly give you an overview of the in vitro systems. Then I'll go over some of the technology features to keep in mind and talk about the considerations regarding the biological resources. And finally, how I believe the MPS program at ATCC can contribute to supporting and enable the field. So ATCC is a nonprofit organization that was founded in 1925. And for nearly a century, ATCC has enabling the advances in science and providing biological resources that enhances the human life and benefits the society. So in the past, ATCC built its collection by cells and microbes that were deposited by research scientists, which continues to happen. But today, we have an R&D team of scientists that are dedicated to developing new products to meet the growing changings of the scientific community. And with that, the ATCC is investing in new technologies such as the MPS. So if we take a look at the evolution of the in vitro models, we can see that they have grown in complexity and functionality. It all started with the continuous cell lines that were isolated from tumors discovered through spontaneous or induced mutation. And next came the manipulation of the cell lines through a transfection or transduction and inclusion of reports. And along with the second generation came the development of protocols for the isolation and preservation of primary cells and specialty media. So on the third generation of the models, I included the gene edit cell line using technologies such as CRISPR, along with the development of immortalized primary cells, which combine the primary cell physiology with the growth properties of the continuous cell lines. And at the current stage, there is a lot of attention of the 3D organotype cultures, such as spheroids and organoids, that are adding layers of improvement regarding the microenvironment. So now we can utilize any of this generation as resources to build models with more complexity, such as the organ-on-chip. So we know that there is a need for models with improved physiological relevance, with better predictability, to be more ethical, to capture the population diversity, and be able to investigate a subset of the population that is often excluded from clinical trials, such as kids, pregnant women, elderly, patients with pre-existing condition, etc. And MPS has been demonstrating improvement compared to the traditional methods. And with so much attention directed to the field, many companies are working on new technologies to improve these in vitro models. And with the boom of new technology, it is not trivial for the end user to select a platform that is suitable for their application. So I have a table of some features that I'm as an end user considering when I am selecting a platform. Having in mind the organ and the application or the question that you're trying to answer, that needs to be recapitulated. There are many attributes that we need to be considering. And one example is the membrane, the pore size and the pore density, the thickness of the membrane can affect cell differentiation, cellular extravasation, cell proliferation, cell projection, and nutrients diffusion. The chemical and physical properties will also have an effect on molecules deposition, absorption, cellular responses, etc. The flow. If the flow is desired, if it is uni or bidirectional, and the platform offers recirculation. So recirculation can avoid the diffusion of secreted molecules, but also if you want to look gradient of the response, it might have a negative impact. So all depends of your question. The material. If you're imaging, and usually imaging, it is desired, it is the model clear. For me, for example, it is. Is the material rigid or soft? It is at gas permeable. Is there a need to adjust for drug absorption? So material-wise also has a lot of a consideration that we have to put it have in mind. Also the throughput. What is the throughput that you desire to have? Usually systems that are extremely complex have a little bit less throughput, and I know that the developers are putting a lot of effort to improve the throughput of their system, but also some of them are already allowing for automation, which is great. And very important, the stimulus. What is the stimulus that the technology offer? And most importantly, if it is in the stimulus in the physiological range that you would like to see. And then many other attributes that you might want to consider, such as an enclosed system, which is excellent to prevent contamination, especially if you're working with infectious disease agents, or if you prefer an easy, accessible platform that allows, for example, for aerosol delivery or easy casting of a gel. So sensor or a technology that facilitates capture of the electrophysiology readouts might be desired if you're working with cardio, neuro, muscle, tissue, for example. And of course, what is the cost associated with the consumables that comes with the platform that are, for me, for example, I know budget, it is something that you have to keep in mind. And very importantly, it is the platform user-friendly. For me, it's critical to have a plug-and-play technology. I have over 12 people that I need to train in various organs in a different context of use. So have something that is plug-and-play, it is important for me. And in summary, there are many attributes to go over. There is no good or bad feature, but what is the question that you're trying to answer? And it's very likely that the end user ended up with a combination of platforms that will complement each other to get to your answer. So independent of the platform of choice, it is extremely important to start with prime biological resources, cell, media, and extracellular matrix. The cells are a very important requirement with considerations to have in mind as a resource for the MPS. Primary cells are a good option since it's the closest to the in vivo physiology and consequently allows for a higher predictability. They do capture the donor-donor viability, that again, it depends on your question if you want to see that or not. But some of the cell types are difficult to obtain due to the various reasons, such as methods of isolation, difficulty in preservation, and also organ availability. They also have a limited lifespan, and in consequence, there is a constant need to assess new donors. Organoids will have a similar composition to the cell of orange and usually has the advantage of keeping the proliferative capacity. But it requires more experienced users, and hopefully automation can overcome this limitation. This tree format, it is already accepted for functional assays, but it can also be used as a resource when we go for organ-on-chip. iPSC have a proliferative capacity, and in theory, the advantage of differentiated in multiple tissues from the same donor. And in many instances, there are limitations regarding poor viability post-op or poor maturation with the presence of fetal characteristics, even post-differentiation. And similar to organoids, it's a cell type that requires more experienced user and a lot of babysitting. but the culture methods have seen a lot of improvements, especially in media and reagents. So I'm hopeful for this cell type. And last, the cell lines. So they are more reproducible and easier to culture, but it might differ from the tissue of orange and might not be fully functional. So you have to check for the presence of your target. So when we are building a complex model, it's very common to end up with a combination of these cell types, depending on what you're looking for or even what it is available. And with the cells growing in co-culture, there is more points that we need to consider. So the model should be validated to demonstrate the cell phenotype and functionality data recapitulates the in vivo physiology in a context of use. It is important to check for the proper differentiated into the major cell types, the presence of the target of interest, when the model is ready to restart an experimental phase, and once you get there, what is the window that you have to actually run your experimental phase? Sometimes you need something that is acute, but if you need something that is a little bit more chronic, you might need something that have a bigger window to do it. Also considering the number of donors and the demographics to have a confidence on your results, which might differ organ to organ or even disease versus health state. Also determine the reagents compatibility when doing in co-culture, since the media and the matrix that are already available for the majority of those cell types, but when you mix them in co-culture, one is going to have an impact and might be a negative impact on the other. So in summary, what is more suitable reagent when it is in co-culture? And in addition, the methods should be reproducible. Sometimes you change the donor and then you have to re-optimize the system. And with all of that to take in consideration, I established the Microphysiological System program to contribute to the scientific community around these biological pain points. So the program currently has 13 scientists working across 4 main segments. The first one it is the primary cells. So this team is responsible for isolating and preserving cells, working together with the cryobiology team, improving the preservation of these cell types that are difficult to preserve, and developing new protocols for additional cell types in characterizing these cells. The STELSAM team is working on developing and characterizing products and checking the differentiation and functionality of the cells. The 3D model block, it is responsible for developing, manufacturing, and characterizing these models. And now the big focus, it is in patient-derived cancer models, which is a big collaboration that we have with the HCMI Consortium. And last, the technology segment. So in this block, we use multiple platforms with various levels of complexity that we are bringing in-house to test our biologicals. We don't want to be biased, so we have collaborating with multiple developers and assess the functionality of our biologicals on the technology. So we often collaborate with the developers to develop new protocols or even new products that can benefit the end users and provide support to the scientific community. So currently the team is working on multiple platforms, assessing the cell lot differentiation, ensure the lot functionality, and working to harmonize some of the protocols across technologies so the end user doesn't need to be training or deal with multiple protocols when using the same organ. So as I mentioned, that sometimes I can see this right now, that we have a lot of technologies that complement each other. And I think the developers do a very good job actually ensuring that we have some tested laws, that we have media and protocol in place. But each one of them have a different protocol. And it's very difficult to be training multiple platforms and multiple protocols at the same time, even when it is the same organ. So what the team is trying to do is focus on having protocols that might be similar across platforms. It might be reproducible across platforms. So our focus on the moment, it is on lung and liver primary models. And you can observe in the first image, these more way differentiated cells on PDMS, on a microphysiological system. You can see the cilia beating in there. In the second image, we have one of the patient-derived cancer organoids that we have continuously releasing through the AGCMI consortium. And at the moment, we have over, I think, nearly 400 models, and we are continually improving the characterization of those as well. The stem cell team is testing the neuroprogenitor cells functionality, but are also working on developing new cardiac models. And all of these teams working together, depending on their organ, on the platform as well, assessing the functionality of the cells. So the big takeaway here is that with so many technologies commercially available, there is a demand for reliable and reproducible biological resources and protocols that are robust and ideally work across multiple platforms with a certain criteria for the application. So I hope that this program can contribute to support this field. I thank you to the organizers and thank you for your time. And please don't hesitate me to contact me if you have any questions or if you have any suggestions or would like to collaborate. Thank you.
Hello, everyone. My name is Ramya, and today I'll be talking to you about immunocompetent models for safety and efficacy assessment. So who are we? So ImmunONE, we're a specialist CRO for in vitro inhalation toxicity assessment, and we have 3 arms. So we're based in the UK. The first is we provide immune-focused immunocompetent models for safety assessments. So we have our own in vitro alveolar in vitro models, as well as we develop new in vitro models according to the need. We provide tailored assessment strategies, so we can provide commercial, we can use non-commercial models as well as commercial models, and it will span all the way from the nasal area to the alveolar region. And on top of that, we provide experimental consultancy. And I would like to highlight that we are experts in high quantum image analysis, and we can use a multiple phenotyping approach to provide safety decision-making support. So at ImmunONE, we don't just like to look at single endpoints for cell death. We like to look at multiple endpoints, which includes immune response. And looking at this, we can predict adverse immune responses at an early stage, which will help mitigate risks. And one of the services we offer is immunophenotyping. So I'd like to talk to you about our patented technologies. This is our immunocompetent models that we have developed in-house. So starting off from the left-hand side, which is ImmuPhage, is a robust and reproducible model of the human alveolar macrophages, and it closely resembles a primary alveolar macrophage in terms of its morphology, biochemistry, and functionality. On the other hand, we have a 3D co-culture model, and it is our human alveolar tissue model, comprising of alveolar epithelial cells and alveolar macrophages in a co-culture setting. So this has the added benefits of ImmuPhage, as well as high barrier properties of over 2,000 ohms per centimeter squared. And essentially, we can use this to provide a detailed understanding of immune and barrier responses in the airways. As well as our products, we are constantly doing different approaches for the R&D pipeline. And one thing is disease modeling. And we're currently looking into fibrosis disease modeling. We're still in the early stages, and especially for idiopathic pulmonary fibrosis. The cells in the models can be raised to air-liquid interface or completely submerged in liquid-liquid. And these are the current versions that we offer, but it all depends on the dosing regime, and we can optimize the models accordingly. Compared to existing protocols, we have found that our immunocompetent model ImmuPhage stays terminally differentiating. And those of you who may not know, our macrophages are not very proliferative, and there is a lot of issues with standardization with existing protocols. And we can safely say that ImmuPhage closely resembles the features of primary human alveolar macrophages. And this can be used as a human relevant tool for safety assessment for inhalation, to looking at toxicity, safety pharmacology, high throughput screening, and even looking at inflammatory studies. So now we have ImmuLung. As I said before, ImmuLung is with the added benefits of ImmuPhage, which I previously just discussed. And the benefits of ImmuLung is the 3D co-culture model. It has a high barrier properties. And these models are standardized and have been beta tested around the world for reproducibility at different CRO sites. And it maintains its barrier functions for up to 14 days in culture. And it's constructed in-house to high standards of quality control to ensure reproducibility and reliable standardized results. Another service that we offer is the in vivo to in vitro extrapolation. And one main issue that's been encountered in preclinical rodent studies is the translation safety concerns. And vacillation and foamy macrophages is something that has been observed in rodents, but no such tool existed to predict such adversity, whether these phenotypic changes in macrophages are safe or if it's adverse response. This leads to key candidates failing and high compound attrition rates. So we worked with the pharmaceutical companies and we wanted to develop and validate an assay to develop a human in vitro tool to assess adverse or safe response of these macrophages, essentially doing a safety profiling. And the way we did this is by using high content image analysis and a multiple parametric approach to provide such answers. So high content image analysis combines alveolar macrophage morphological endpoints as well as lipid endpoints as an in vitro tool, and it can be used as a preclinical tool for inhaled safety assessment. And the morphometric endpoints that is obtained is directly correlated with that you would find in vivo rodent studies. So how did we do the study? So we have a publication on this also, which I'm happy to share after. And so we use rat and human alveolar macrophages. We dosed them with a panel of 12 compounds. Half of these were foamy macrophage inducers. The other were non-inducers, so they didn't cause any foaminess. We dosed alveolar macrophages, and the cells are stained with a dye cocktail, with fluorescent dye cocktail, looking at different regions of the cellular regions, such as looking at mitochondrial activity, cell membrane permeability, lipid uptake, nuclear area, and vacillation profile. So we used the quantitative high content analysis approach to look at cell health parameters and morphometric analysis. And the benefit of this tool was we were able to look at the morphological characteristic and measure simultaneously in a multiplexing fashion. Now we have a case study where models, our immunocompetent models are applied and compounds were exposed and trialed. So the problem. So Company X approached us with small molecules and they had some pre-existing in vivo data and they wanted to seek to address some main concerns. One of them was model validation. They needed us to confirm if our in vitro models could replicate the adversity that they've seen in the rodent studies. And essentially, they wanted a tool that's cost effective rather than using a lot of money and for it to fail later down in the pipeline. Another factor they wanted to look at was early assessment. They want to see if this in vitro tool that we can provide early mechanistic insights into safety of these molecules and essentially, like I said, potentially saving costs by avoiding late stage failures. And another mandate was looking at immunology. So one of the things that they observed was cytokine changes in the preliminary rodent studies, and these pro-inflammatory markers that were present, it was an important endpoint to be measured for this study. And I think it was a mandate in their regulatory submission. And as there was limited availability to companies to do such areas in deep lung for immune responses and conducting high content image analysis services, they kind of reached us to do this for them. So what was ImmunONE's tailored solution? So we provided a phase study approach, study A to E, starting with MORPH1, which is the morphology assessment, and looking at cell health parameters using a high-quantity image analysis. We then moved on to look at the bit of the functionality, looking at phagocytosis, lipid uptake via flow cytometry, and studies see looking at we wanted to look to see if there was any change in polarization states, and cytokine release was looked at, and finally the barrier properties. I will now showcase some of the results that we obtained. I will just focus on some of the findings. So this is the MORPH1 assay, and on the left-hand side, as you can see here, it's like a heat map from the high-quantity image analysis. It's like a barcode to kind of, this is how we phenotypically profiled them. And on the right is all the images from the high-quantity image analysis. In order to do this analysis, we use 4 descriptors, such as mitochondrial activity. We looked at the vacuolation profile and the extent of vacuolation. And we use these descriptors to give insights on how cells respond to drug of interest. And from this, we got 81 different phenotypes. And it was interesting to see that our findings correlated to that the company X found in vivo. And some of the findings, so the ones highlighted in the red boxes, some of the findings were upregulation of mitochondrial activity, cell area, vacuolation profile, and it's a bit of the shift in the barcode, as you can see. And what this kind of tells us is that this tool, high quantity analysis tool, using a multi-parametric approach, is quite quick and efficient and can be used as a tool to assess compounds very early, and we can fail your candidates early in the pipeline. And this is some of the cell health parameters that we looked at: cell count, membrane permeability, mitochondrial activity. I'm only showing you cell count and membrane permeability graphs here. And from compound C, which I showed earlier, which was causing a shift, we were able to take out this information, and you can see that compound C decreased the cell count and had the ability to increase the membrane's permeability. And this was based on the average population data. And Imiphage was able to show that compound impacted the characteristic parameters of the cells. Now, looking at cell morphology parameters. So some of the morphology parameters we looked at was nuclear area, cell area, area of vacuoles per cell, percentage cell area, and the extent of vacuolation. And I'm only showing you the graph for nuclear area. And morphological changes were observed with compound C. Nuclear area was upregulated for compound C. No changes to cell area. There was decreased vacuolation per cell, decreased percentage area, and the extent of vacuolation. And it's important to note that changes in nuclear area is directly correlative when we're trying to understand toxicological findings. Study B looked at the phagocytic activity and lipid uptake. So on this, I've labeled engulf 1. This is our phagocytic activity assay, and lipid 1, as the name suggests, looks at lipid uptake. Intracellular lipid content and phagocytic data. So compound A was increasing phospholipid content. There was no change with B observed. Compound C, on the other hand, was downregulating intracellular lipid content. And when we're moving on to engulf 1, which is a phagocytic activity, compound C and D decrease phagocytic activity. And in correlated toxicity, looking at phospholipid content and its ability to phagocytate the inhaled products is quite important. This is just a quick overview of the macrophage polarization assay we've done. And we found that the compound A that we took forward was able to shift the macrophage polarization towards an M2 state. The M2 macrophages are kind of involved in homeostatic pathways. And it wasn't the CD163 marker that we measured, it wasn't as significant as the M2 macrophage control, but there was an elevation in that marker. Now moving along to cytokine and chemokine analysis. So this is based on a human protein profiler cytokine array assay over 24 hours and 48-hour dosing. And from the samples tested, so there's like a membrane arrays, they're spotted with different cytokines. And we were able to detect 16 cytokines and chemokines from the samples. And you can see that, so in terms of the red, I would like to say that it's upregulation, and anything in the blue means downregulation. So upregulation of pro-inflammatory markers was observed for compound C. So this is just data from compound C, and it shows this is data from A to D, and compound C shows that it elicited a pro-inflammatory response. And this is interesting to see, as this finding was also observed in vivo by company X, and a similar response was seen for compound D. Finally, we looked at the epithelial barrier properties. So compounds A, B, and D had no significant effects, whereas compound C disrupted the barrier properties. So in terms of take-home findings, what were we able to find out? In terms of compound C validation, compound C elicited pro-inflammatory changes and macrophage alterations, and this confirmed its consistencies with in vivo finding that company X originally found. This suggests the immunophage platform can be used for in vitro extrapolation studies. On the other hand, molecules A, B, and D, we were able to provide early insights. While there were changes and alterations observed for cytokine expression, they showed no macrophage morphology changes, and this kind of indicated to us that they're likely safe. This is based on the benchmarking against safe and unsafe compounds using the high content tool, and this is the findings that we were able to provide for them. So in summary, ImmuPhage and ImmuONE are great human relevant tools for safety decision making. In terms of high content image analysis, derived profiles, you're able to effectively differentiate between safe and unsafe macrophage responses. And rather than just looking at single endpoint, I believe that a multiple parametric and analysis is essential for early decision making. And from this, I would like to thank you for the opportunity, and I welcome any questions. Thank you.
Hi, everyone. First off, I would like to thank the organizers for inviting CuriBio to participate in the workshop today. I'm Sean Luttrell, Product Manager for Cell and Tissue Models. And today I'm going to talk about one of our cell models that we developed and commercialized for skeletal muscle tissue engineering. And before I get into that and show you that data, I'll give you a brief overview of our company and a couple of our key platforms that we use to characterize our cell models. So first off, CuriBio, we are a biotech company in Seattle, Washington. Our platforms integrate biology and engineering into a preclinical pipeline to inform R&D decision making. We do this with three primary sets of technologies and tools. We develop advanced human 2D and 3D tissue models for disease using induced pluripotent stem cells. We build proprietary biosystems that we use to measure clinically relevant functional data from these biological models. And then we use AI and machine learning to analyze the data from these tissues and biosystems in a more high throughput approach. And our goal, of course, is to create human in vitro models and platforms that better replicate in vivo biology to improve the translatability of preclinical testing. And we believe that one way to better model human biology is by using 3D muscle organoids with cells optimized for tissue engineering. And so this is really one of the core products that started our journey into 3D tissue engineering. This is the ManoRay 24-well plate featuring 24 strips of contractile tissues. This is a two-post system with one rigid post on the right and a flexible post on the left with a hydrogel containing cardiac or skeletal muscle cells within a matrix in between the two posts. Inside that flexible post is a high-powered magnet, and that's the black square that you see. Every time the tissue beats or contracts, the magnet moves, and that's essentially how we measure contractility in our instrument. We developed this technology because we believe in the scientific benefits it provides. So compared to animal models, you now have human-derived tissues with the clarity and ease of control of in vitro experiments. And compared to 2D cultures, you have improved physiological relevance. So you are now unifying chemical and mechanical cues in the form of extracellular matrix, within which different cell types can interact with the ECM and with each other. You can have extended culture times in these tissues. So 3D tissues can easily last for many, many months, basically as long as you can keep them sterile. And this allows for long-term disease modeling and therapeutic studies. And then we now have access to clinically relevant functional data in the form of output, as one can measure the contractile force and kinetics over time, and in the form of input as electromechanical conditioning for maturation or injury of the tissues. And so to make these tissue casting process as easy as possible, we designed a casting consumable so you can create tissues using basic cell culture skills, but without the typical shortcomings of these 3D models, such as low throughput, low reproducibility, and then time-consuming engineering procedures. So here is how the casting procedure works. This is a tissue casting consumable that we designed. It's a modified 24-well plate. The upper portion is a lattice that holds 24 sets of posts, one rigid, one flexible for every well. In the bottom of each well of the casting plate is a trench, and you can see a close-up of the trench at the top. This is where you will fill that trench with your cell hydrogel components, pop it in the incubator, and 80 minutes later, you have 24 mini tissues that will be attached to those posts. You can put these tissues into a new plate of media, and heart tissues will start to spontaneously beat after 2 to 4 days. Skeletal tissues will contract under stimulation after about 5 days. So in this system, we have about 95% casting success. And after the tissues are contractile, then we can measure function. And for this, we use our manta ray instrument. This is about the size of a shoebox. It's inside of a standard cell culture incubator. And there's an array of magnetometers under this plate of tissues. So when we use a custom stimulation lid to induce these tissues to contract, the manta ray can measure movement of every flexible post in the 24-well plate in parallel and in real time, generating a force versus time waveform of contractility. And this is where we can derive all of our contractile metrics from our tissues. So this is a very basic overview of how we make our tissues and we measure function in terms of contractility. And so now we can use this system to validate contractility metrics in our cell lines and disease models that we produce. So the first step in making our disease models is producing the cells. And for that, we start with iPSCs, and then we use a directed differentiation approach to generate myoblasts after about 30 days. We enrich these cells using FACS to increase our myoblast purity, and we can confirm the purity using flow to test for Desmond. After the cells pass our initial quality control metrics that I'll describe in a couple slides, we can expand our enriched cells, bank them in high-density cryovials for 3D functional assessment and tissue engineering. And when we're ready to actually make the tissues, we can thaw a single cryovial and cast those cells directly into our casting plate for 3D functional validation of the cell lines. And since we're using the same lot of cells over many plates, this improves the consistency and the repeatability between tissues and between different experiments using these cells. So the first disease model we developed and commercialized at CuriBio was an isogenic pair of iPSC-derived myoblast lines modeling Duchenne muscular dystrophy, or DMD. And this is the data that I will be showing you today. Let's see, so DMD is the most common form of muscle dystrophy. This is an X-linked disease, and it primarily affects young boys. Symptoms start in early childhood with difficulties walking, running, and climbing. And then as the disease progresses, children are usually wheelchair bound by age 12. Few patients survive into their 20s. And now with improved ventilatory support, cardiomyopathy is now the leading cause of death in these patients. There currently is no cure for this disease, and thus there is an urgent need to pursue new strategies to prevent, halt, or reverse disease progression in DMD patients. So this is a rare muscle disorder. It affects about 1 in 3,500 male births. It's caused by mutations in the dystrophin gene. This is the largest gene in the genome. Mutations are typically deletions or duplications, And dystrophin protein plays a very important role in the cell. It links the contractile machinery to the membrane. And so essentially it's acting as a shock absorber or stabilizer during muscle contractions. So to model this disease, we CRISPR engineered a DMD line with a deletion of exon 45 and a 17 base pair deletion in exon 54, leading to a premature stop codon using iPSCs from a healthy adult male. And this is the model I'll be describing to you today. So the first step in validating our model is to run a few quality control checks in 2D. And so the first thing that we want to test is viability out of thaw, purity of the cells, and then also myoblast fusion. And so here on the left, you can see a general morphology of both myoblast lines. These cells are quite robust. They have a typical viability of about 95% or better out of cryopreservation. And this is one reason that we can put these cells directly into our 3D tissue models out of thaw without any 2D culture or additional expansion. So the next thing we look at is cell purity in 2D. And this is just a representative figure here showing you that we use flow staining for Desmond to quantify the percent Desmond positivity, but the actual purity of both of these cultures is, or both of these cell lines is here in the corner. So above 80% for both, and that is our threshold for purity with where we want to be for tissue engineering. And this ensures when we're, especially when we're comparing different cell lines, and definitely with isogenic lines, we want to ensure that we are incorporating roughly the same amount of myoblasts into our tissue constructs. So when we're making comparisons in function between the two lines, we can be relatively confident that they both contain the same number of muscle cells. And so the final metric that we look at is fusion. And we want to be sure our myoblasts are fusing together to form multinucleated myotubes. And this is more of a qualitative measure since quantifying a fusion index is nearly impossible in my experience. But here we want to see a monolayer of contractile myotubes after just a few days in our differentiation medium. And so at this point, these myotubes start to spontaneously contract and combined with passive tension that they form as they develop, they typically will delaminate rather quickly from these flat surfaces. And so to quantifiably measure function from our cells, we incorporate them into our 3D muscle tissue organoids. And this is where we can easily validate function over an extended period of time. So the first thing we look at in our muscle bundles is force over time. Here we've made the tissues and we've tracked function over 43 days. So this is what they look like at day 3. And you can see they continue to compact as they are maintained in culture. And so these tissues do last for quite some time, as I said, they'll remain relatively functional for as long as you can keep them sterile. And this really enables long-term therapeutic testing or disease modeling. And so if we look at morphology, we see the tissues do get darker with time. And this indicates that compaction of the hydrogel is happening as the cells are remodeling the matrix. And so we do include 10% fibroblasts in our cultures, and this helps to stabilize the tissues over extended culture times. When we look at the twitch force of these muscles from day 5, we see the tissues are quite strong through early development, and then they reach a fairly steady homeostasis over the first month and a half of culture. And so here in this model, green is our healthy control cell line, and DMD is our isogenic disease line. And you can see that the control cell line is much stronger than our disease line. So there does seem to be a disease phenotype, at least in terms of force. So if we look at a single day in time, a very clear disease phenotype starts to present basically from the start of 3D culture in our model. So these are data from day 10 tissues. At the top of the slide, you can see tissue morphology to the eye is quite similar. But when we compare them at the functional level, we see a significant deficit in our DMD line. Twitch and tetanic force are 3 to 4 times weaker in the DMD tissues. And here's an example of the rich information that we can get from contractility measurements on the man array, particularly in the disease modeling context. So you can see that DMD disease model stratifies across many different components of contractility. All force and kinetics measurements are statistically different between the isogenic pair. And this really gives us a wide range of metrics to assess effect and recovery with different therapeutics aimed at treating different aspects of the disease biology. Fatigue is another hallmark symptom of DMD, and this is a great example of a clinically relevant aspect of the disease model. When we stimulate the muscles over a 30-second time frame here, we're using a twitch stimulation protocol. You can see that the last contraction is producing less force than the initial contraction. So if we plot each of these force values as a percent of the initial force, we can measure our fatigue rate. And you can see our disease line fatigues much faster. And so you can start to ask questions like, can this drug improve fatigability of the muscle? You can also couple this with other measurement techniques to determine things like damage of the muscle. So the ManArray platform enables us to assess muscle function in very complex ways. Aside from just simple twitch and tetanic stimulation, we can design stimulation programs that test the ability of the muscle to move calcium and contract and relax appropriately in response to the type of stimulation we deliver. We do have a custom well-based stimulation lid, so we can stimulate one tissue at a time, 24 tissues. We can use 24 different stimulation protocols if we wanted, and then record the resulting contractility across all 24 tissues in parallel. And so I really love this slide because it so beautifully shows how our in vitro data is a mirror image of in vivo muscle function. We ran a force frequency in these tissues, and as we increase the frequency of stimulation, we see a corresponding increase in force of the muscle. And so we see a very discrete changes going from twitch through summation and all the way into a fused tetanus. Furthermore, we can see a statistical difference between our disease model pair that looks strikingly similar to a healthy and DX mouse model of Duchenne muscular dystrophy. And so if we plot each one of these force values over each frequency, we get a nice graph that clearly shows the statistical difference in function between the isogenic cell lines.
Sean, just to let you know, you've got about a minute left.
Okay, we can fix these tissues. We can run histology to look for protein localization. Importantly, our DMD line does not express dystrophin, so that's important. And then finally, we can use different compounds to see if our tissues are responding as they should to different drugs. So BDM is an ATPase inhibitor, so it should shut the muscle down. When we apply it to our tissues, it does just that. We can wash it out and force return, so we know we're not just killing the cells in the tissue constructs. We can also increase force with a troponin activator, tirasemtiv, and this was just a pilot study, but you can see at these two doses, we do see an increase in force, showing that the drug is having the effect that it's supposed to have. And finally, I'll just briefly say that the function that we get out of these tissues is greatly dependent on the media that we provide. Since we do have a culture of fibroblasts and myoblasts, it's very important to give them a media that supports both tissue types, but also prevents unwanted effects of tissue compaction and remodeling. And so our media, when compared to the leading competitor media, and then also a conventional medium, which has been well described in the literature, outperforms significantly across all of these areas, as you can see. So you can buy our cells, you can buy our media. We provide our media to you, depending on the application used, whether it's 2D or 3D tissue engineering. You can provide different formats of cells in 1, 6, or 12 million cell cryo vials. We have a new model that we'll be offering soon of DM1. This is a 3-cell line model going from a severe phenotype to a less severe phenotype to a corrected control. And we hope to get that to you in the next coming month or two. So with that, I'll just say you can reach out to us and work with us in a variety of different ways, purchase our instruments, work with us as a CRO, partnerships, consult. You can license some of our models. And I think I'm hopefully right at the edge of my time, I would thank you and happy to take questions at the end of the workshop.
All right. Hello, everyone. First, thank you for the opportunity to present here today to you, part of our data. My name is Nick Nicolaou. I'm the head of R&D at DefiniGEN. And what I'm going to show you over the next 12 to 15 minutes is some recent data we have generated over the last 2.5 years, demonstrating the quality of our product, which is iPSC-derived hepatocytes, and how this could be used for drug efficacy and toxicology screening. A few words about us. We are a spin-out company from the University of Cambridge. We are relatively small. We are about 20 people in the lab and outside the lab. However, we have more than 10 years of iPSC-derived liver cell expertise. Actually, our lab was set up at the University of Cambridge, as I said, from Professor Ludovic Valliere, and our proprietary protocols focus on the generation of liver cells focusing on coming from iPSCs. And based on that proprietary protocol, we have established a variety of wild type and disease liver cell models, and we are ISO certified. And why do we exist? Actually, what we're trying to do is to address the challenge. And the challenge focuses on the high need of efficient and relevant in vitro liver models that can be incorporated into the drug discovery pipelines and identify potential drugs and their toxicity profiles. However, the current liver models that are used in drug testing, in liver drug testing, are often rely on cancer cell lines, hepatocellular carcinoma cell lines, including Hep-G and Hep-G2s, or primary human hepatocytes. And both of these models have major limitations. And this is where DefiniGEN comes. So what we're trying to do is to bridge this gap and generate a model and a platform that can be optimal for both drug efficacy and predict toxicology screening. But what are the models that people are currently using in liver research nowadays? As I said, the gold standard in vitro liver cell model is primary human hepatocytes. And of course, many of you that are related to the field know that they are really good cells. When they are in culture, they can retain metabolic activities. They have drug transporter activity, and you can use those cells for drug toxicity characterization studies. However, they come with significant disadvantages as well, and this is actually the rapid loss of metabolic function once you put them in culture. There is huge uncontrolled patient-to-patient variability and batch variation. Quite frankly, there's no wild-type control because all of them usually come from liver resections, and this modeling, quite frankly, is rather not possible. Because of those problems, people are using instead hepatocellular carcinoma cell lines, as I said, Hep-G2 or Hep-Gs, and these cells are quite handy. Liver biologists, including me, have used these cells in the past. And they're very handy because they are very cheap and they have great expansion capacity and phenotypic stability. However, they are carcinoma cells, therefore they are not physiologically relevant. They have an abnormal karyotype and they lack crucial metabolic pathways that are important for both metabolic studies as well as disease modeling. And this is where we come at DefiniGEN, where what we're trying to do is to take into account all the advantages of both liver cancer cell lines as well as primary human hepatocytes, in particular the expansion capacity and phenotypic stability of cancer cell lines, and combine that with the functionality and healthy karyotype of primary human hepatocytes. And as I said, what I'm going to show you from now on is some crucial data that we have performed over the last few years, showing that our cells perform all the major metabolic liver functions, including albumin production and secretion, carbohydrate, aka glucose metabolism, urea cycle or protein catabolism, bile acid homeostasis, and drug detoxification. But first, let's see how the cells look when you put them in culture. On the top part of the slide, you can see our differentiation protocols, how we start from iPSCs, and following our proprietary protocol after 2 to 3 weeks, we end up in a very nice monolayer of iPSC-derived hepatocytes, or OptHep, as we call them. This is the brand of our company. And they're very nice. They're polyhedral. They have that cobblestone-like morphology that you can see when we zoom in, which is very similar to what we have with primary human hepatocytes. And in no way they are similar to Hep-G2s because they are cancerous, they are epithelial-like. So by default, we know that our cells are even morphologically better compared to the liver cancer cell lines that many people are using and very similar to primary human hepatocytes. Moving forward to the characterization, the gold standard characterization when it comes to iPSC-derived hepatocytes is actually investigating whether those cells express, the standard liver maturity markers, including albumin, alpha-1 antitrypsin, and hepatocyte nuclear factor 4A. And this is what we have done at DefiniGEN, similar to many other companies and labs. But what we have done on top of it is actually to compare those levels to primary human hepatocytes, PAH, as well as HepG2s. And as you can see from those immunocytochemistry-related pictures, our cells express high levels of albumin, similar to what we observe in primary human hepatocytes and HepG2s. Here, actually, in this graph, you can observe that we have more than 90% positive cells expressing albumin, similar to what we observe in primary hepatocytes and HepG2s. I would like to point out here that for color coding, from now on, all of our data will be in blue, showing the data from our cells, our OptiHeps. In pink, we have the primary human hepatocytes, and in purple, we have the HepG2s. Our cells also express high levels of HNF4 alpha, similar to what we observe in primary human hepatocytes and in HepG2s. And they also express alpha-1 antitrypsin, a liver maturity marker, which is quite important, both because it demonstrates the superiority of our cells compared to HepG2s. We have no expression in HepG2s. And of course, because we can use those cells for liver disease modeling, including alpha-1 antitrypsin deficiency. But what I would like to point out here is this fourth marker, alpha-fetoprotein, because as some of the presenters mentioning earlier, iPSC-derived hepatocytes in the past have been quite unpopular and infamous about representing a fetal hepatocyte stage. Therefore, they were not able to demonstrate a mature hepatocyte functionality, and that was making them not comfortable and not convenient for liver studies. But not with our cells, because as I said, over the last few years, we have extensively characterized and optimized our differentiation protocols. And now we are happy to share that our cells have escaped that fetal hepatocyte stage, and this is quite nicely reflected by the really low levels of alpha-fetoprotein, and these are actually comparable to what we observe in primary human hepatocytes and HepG2s. And we didn't stop there. We also wanted to show how much albumin our cells secrete and how that compares with primary human hepatocytes. Here you see a time-dependent secretion study showing that our cells, that by day 9 following differentiation, they secrete comparable levels of albumin to what we observe in primary human hepatocytes. And when we move to day 12, our cells start secreting even more when the primary human hepatocytes start dedifferentiating. And this is consistent with the rapid loss of metabolic function that those cells show in a 2D monolayer. But what was really nice is that this day 12 bar here is actually this day 12 bar here, day 12. And what does that graph show? That graph shows actually that we can keep the cells for at least another 2 weeks in culture without losing their phenotype. And this is a great advantage when it comes to iPSC-derived hepatocytes, because you can keep our cells in culture for more than 3 weeks, first, without losing the phenotype, and second, giving the opportunity to perform long-term studies and experiments. And that was consistent with the second metabolic pathway that we really wanted to investigate. And the pathway is focusing on the urea cycle, which is actually focusing on the detoxification of the toxic ammonia towards urea. It's a liver-specific pathway. For those who are not very familiar with the field, it's a liver-specific we have liver-specific enzymes that express either in the cytoplasm or in the mitochondria, and you need all these 5 enzymes in green to have a fully functional urea cycle. And this is what we investigated in our cells by performing Western blot data here. We saw that our cells express those enzymes in high levels, way higher to what we observe in HepG2s. And these protein data were very nicely reflected here in this graph B, which shows the high levels of urea secretion that we have in our cells and how much higher they are compared to what we observe in HepG2s. And similar to the albumin secretion data, we see that our cells remain stable when it comes to other metabolic markers, in this case, urea. And you see that by day 20 following differentiation, our cells have maximized the urea secretion ability, and they can remain stable for a period of time, again, allowing long-term studies. The third pathway that we were really interested to investigate as liver biologists was the de novo gluconeogenesis pathway. This is, again, a liver-specific pathway, and it is based on the ability of liver cells to generate glucose at states of fasting. At this side of the slide, I have put a simplified schematic of the pathway. It is a rather complicated pathway, but what I would like you to focus on is on the rate limiting step of the pathway, and this is glucose 6-phosphatase, and how this is converts glucose when we have pyruvate as the initial substrate. For us, the first challenge was to investigate whether the rate limiting step was actually expressed in our cells. And for this reason, we performed qPCR experiments, and we compare the mRNA levels of G6PC in our cells in blue, in primary human hepatocytes, and in HepG2s. And you can see that our cells expressed higher levels of HepG2s and more closely related to what we observe in primary human hepatocytes. But what was really nice was that this pathway does not only show expression of G6PC, but it is very sensitive to amendments. So what we did, we tried to induce this pathway by treating our cells with gluconeogenesis inducers. In this case, we use the gluconeogenesis inducer DPCAMP. And what we found was a marked increase in the expression of G6PC in our cells in blue, and the impaired increase of G6PC in HepG2s, again reflecting that these cells are not the best model when it comes to metabolic studies. And finally, when we came into the product, which is actually glucose, what we did, we actually treated our cells with increasing concentrations of pyruvate. The hypothesis here would be that the more pyruvate we add in our system, the more glucose we generate. And indeed, this is what we observed in our cells. We observed a dose-dependent increase in glucose secretion when, again, HepG2s did not show any stimulation with regards to the de novo glucogenesis pathway. And now I'm moving to some of the most recent data that we have generated in our labs, and these actually focus on the potential ability of our cells to be used for drug toxicity studies. Because again, in the past, iPSC-derived hepatocytes were failing to demonstrate cytochrome P450 expression profiles, but not anymore. Following the optimization of our differentiation protocols, as I mentioned earlier, we investigated whether our cells express the standard, the major CYP450 enzymes that are needed for drug metabolism within the liver. And here you see the mRNA panel where we check the expression levels of CYP3A4, 2B6, 2C9, 2C19, 2A6, and 2J2. And the levels of those markers were actually comparable to what we observe in primary human hepatocytes and way higher to what we have in HepG2s. But as a company, and as I have shown you already, we are not only interested in mRNA levels, we are mostly interested in functional activity and protein levels. And this is what this graph shows here. This graph shows the basal CYP3A4 activity levels in our cells. And actually you see that these levels are comparable to what we observe in primary human hepatocytes. And again, way higher to HepG2s. And I would like to point out that these primary human hepatocyte data is the result from 3 independent donors. So we have extensively characterized that model as well. And our cells not only express CYP3A4 and have basal activity, we can actually induce CYP3A4. In this case, we induced CYP3A4 mRNA expression by treating our cells with 2 different CYP3A4 inducers. In this case, it was rifampicin and vitamin D. And you see a more than 6-fold increase in CYP3A4 mRNA levels when the cells were treated with rifampicin, and more than 17-fold increase when the cells were treated with vitamin D. And these mRNA data were very nicely reflected to the protein data, to the activity assay. What we actually did here, this is a rather complicated graph, was to treat our cells with 2 different concentrations of the vitamin D inducer, 10 nanomolar and 100 nanomolar. And as you can see, we performed the same experiments in both primary human hepatocytes and HepG2s. And what we observed was that actually our cells could have a similar response to primary human hepatocytes with increasing concentrations of the inducer. And on top of that, when we actually co-treated our cells with a CYP3A4 inhibitor, in this case ketoconazole, the co-treatment of both the inducer and the inhibitor was able to reduce and rescue CYP3A4 protein levels, showing in total that our cells have CYP3A4 activity, they have basal activity expression, they have basal activity, we can induce and we can actually inhibit that. And finally, having shown that our cells have those phase 1 gene expression profiles quite active, we wanted to investigate whether our cells can be used for drug-induced liver injury related studies. So in collaboration with AstraZeneca, we treated our cells with a panel of 25 to 30 drugs of known liability. Here we are presenting 7 of them for the interest of space and time. But what we have did to make it easier for the audience is we have split those drugs based on their DILI concern. So in shades of blue, we have the drugs of high DILI concern. In shades of green, the drugs of green, the drugs of medium DILI concern, and in shades of red, the drugs of no DILI concern. And when we actually performed cell viability, cell toxicity studies with increasing concentration of those drugs, we observed that the high DILI and the medium DILI concern-related drugs resulted in increased cell death with increased concentrations, when the no DILI-related drugs had no effect on cell viability. And that was quite consistent when we checked an additional metabolic marker. In this case, we used albumin secretion. And here you see that the no DILI concern-related drugs in red have comparable levels of albumin secretion to the vehicle-treated cells. However, the high DILI and the medium DILI concern-related drugs resulted in reduced albumin secretion, again validating that our cells can be used for drug-induced liver injury prediction studies. And finally, we wanted to investigate whether additional efflux transporters that are quite important for both bile acid metabolism as well as drug-induced liver injury. In particular, I'm focusing on the ABCB11/BSEP transporter. We know that BSEP is now considered, BSEP inhibition is now considered a DILI risk factor. And for this reason, we wanted to investigate whether our cells express and localize that transporter properly. Again, we compared our cells to primary human hepatocytes and HepG2s, and as you can see from this mRNA graph here, our cells had comparable levels of ABCB11 to primary human hepatocytes when HepG2s do not express it at all. And the same pattern was observed with other efflux transporters, in this case ABCG2 and MRP2. But what was quite nice for us was that our cells did not only express it, but they localized that properly. Here you see some representative immunocytochemistry-related pictures following sandwich cultured iPS-derived hepatocytes, where you see the formation of really nice bile acid canaliculi and the proper localization of ABCB11 in the hepatocyte membrane. To conclude, I hope that I have shown you that current in vitro liver models come with significant limitations, and that our cells can demonstrate all aspects of liver cell function, including hepatocyte maturity marker expression, urea cycle, de novo gluconeogenesis, C450 activity, induction and toxicity predictivity, and efflux transporter expression localization. In total, we are hoping that our cells are superior to the existing preclinical models. They can provide highly predictive in vitro models for drug efficacy and toxicity studies, and they can pave the way for a more efficient and effective future in the field of drug discovery. I would like to thank you for your time, and I'm happy to take any questions either now or later during the Q&A.
Excellent. So thank you for everyone for joining me today, and thank you to the 3Rs for putting together this supplier-focused session. My name is Emma Rogers. I'm the Laboratory Director of Obatala Sciences, and I appreciate the opportunity to speak with you all today. I hope to provide an introduction to our product catalog as a whole, as well as discuss how our products can enable your overall research objectives. I'll end by discussing opportunities for collaborations and other partnerships with groups in this industry space. So if you're not already familiar with Obatala Sciences, we are a small biotechnology company located in New Orleans, Louisiana. We are a partner in developing human biological models and research tools for drug discovery applications. And like many of the other organizations here today, we're in business because the use of animal models and animal-derived biomaterials in preclinical research is detrimental to clinical translation downstream. Secondarily, even when humanized models are considered in this context, they often do not incorporate representative or diverse patient populations when considering study design, human sample origin, and the use of human biomaterials. For diseases like diabetes, obesity, cancer, heart disease, and even correlated pathophysiologies like wound healing, the lack of diversity in research has impeded the development of therapies that are accessible, efficacious, and equitable for patients worldwide. Here at Obatala, we address these problems by developing human tissue models from stem cells and biomaterials to better recapitulate human physiology and by taking into account human diversity. We recycle routine medical waste from elective surgeries with patient-informed consent, From various sources of tissue, we isolate stem cells and other human biomaterials. We combine all of these materials to further develop complex, physiologically relevant human tissue models that are typically used for drug screening and drug discovery applications. Our catalog includes a biobank of human stem cells from multiple tissue sources, including primary cell lines like the stromal vascular fraction, a heterogeneous primary cell line with 8 different subpopulations that we've characterized extensively. Our hydrogel products are derived from human tissue sources, and additionally, we've demonstrated that our hydrogels are tunable, they support multiple cell types, and are compatible with multiple culture endpoints. Our media product line includes growth media, for which we have a xenoprotein-free variation in the pipeline, as well as differentiation medias to support 2D and 3D cell culture. That's on small scale, large scale, bioreactor scale, all of the above. Our tissue chip kit combines all of these materials as a kit to enable ease of use and quick setup in our customers' hands. And then additionally, our live plates are available as a product that's shipped to you or as a service. They're compatible with multiple culture formats and culture endpoints. Our live plates include a 3D model for human fat on a chip, as well as obesity on a chip. In the live plate format, these are non-perfusion systems. They can be adapted to fit your needs. Our products, all of the above, can be purchased from our website, also available on distributor platforms, and the number of distributor platforms where our products are supported by are growing every day. And you can also email orders at obatalasciences.com, and I'll have a closing slide at the end with more of that contact information. So this is a snapshot of our cell inventory to illustrate our focus on diversity. Our biobank includes characterized samples from over 200 donors. That number is growing every day, for which we retain de-identified donor demographic information as well as disease history. For a given donor, we inventory multiple tissue types. Within a given tissue type, we may receive several depots, all of which we are able to retrieve cells from. We develop custom donor pools, matched donor pairs to investigate variables like age, sex, ethnicity, as well as BMI, and matched tissues to investigate other variables of interest. We evaluate all of our cells against minimum quality standards for commercial release. For primary cell lines like the stromal vascular fraction, we assess sterility and viability at isolation. From there, we characterize our stem cells at early passage numbers, that's passage 0 whenever possible, to assess key markers of stemness, namely proliferation, trilineage differentiation, clonogenicity, and immunophenotype. All of this is captured in the sample C of A at right for a given donor lot. This ensures that the vial of cells that our customers receive has been evaluated for viability, live cell number, post-thaw, as well as functionality, a central tenet of good cell culture practices. With good cells, of course, necessitates good media products. Our media products include StromaQual, a growth and expansion media, for which we are launching a xenoprotein-free variation later this year, as well as Adipoqual for adipogenic differentiation, OsteoQual for osteogenic differentiation, and ChondroQual, our chondrogenic differentiation media. All of these products are available as 2D and 3D formulations to support a variety of culture formats, culture scale, and things like that. We also offer bulk rate and subscription-based purchasing options to facilitate all of our customer needs. On the other hand, our biomaterials are at the core of our technology and highlight a critical need for not just matrix products that are animal protein-free, but also products that are representative of human tissues. Our line of human-derived hydrogels are sourced from a variety of tissue types to better recapitulate human microphysiology and native tissue architecture. Outlined here is the stiffness profile of our human blood-derived hydrogel Obagel, as compared to its ECM-rich counterpart, aptly named Obagel ECM, for which 2 different dilutions are shown to illustrate tunability. ECM is a tunable matrix that is compatible with soft and hard tissue modeling applications. Obagel really suits that very soft tissue modeling space. These hydrogels are stable in frozen storage. Once thawed, they retain liquid status at 4 degrees to just below room temperature, while gelling at 37 degrees by body temperature. Gone are the days of your matrix gelling in the vial or the pipette tip. I've experienced both. Notably, we have established quality assurance measures to ensure lot-to-lot consistency, which is also often seen as a major drawback of competitor products. The figure above shows stiffness for 2 different lots of Obagel ECM, as compared to 2 lots of Corning's Matrigel product. To briefly highlight a few more of our strengths, we are proud to say that Obatala Sciences is ISO 9001 certified as of last year. We perform extensive quality assurance and quality control measures for the release of commercial products, cell lines, biomaterials. A sample workflow for hydrogel manufacturing QA/QC is shown above, from point of origin with donor tissue samples through controlled manufacturing processes, quality testing, and commercial lot release. Additionally, our SOPs implement good cell culture practices, good laboratory practices for methods that are published within the context of our core expertise. And then again, by design, our products support multiple MPS formats and applications, which we view as a real asset. We have demonstrated the use of our biomaterials in microfluidic chips, including vascularized chips, multi-layer perfusion platforms, and perfusion bioreactor platforms. Additionally, I've highlighted a PDX study performed with a collaborator in an immunodeficient mouse model for lung adenocarcinoma, showing comparable tumor volume over time for scaffolds of interest as well as Obagel. We are looking for additional opportunities to partner with industry and academic groups for bioprinting and bioink applications, cancer modeling, and incorporation of adipose tissue into functional organ models and multi-organ on-chip systems. The ways that we typically partner include direct sales, service contracts, collaborations for grant applications, as well as supporting research to further advance publications or even beta testing applications. That's your products, our products combined. Any opportunity that gets my hands back in the lab to do beta testing applications is a happy day. So again, I'm thrilled to have the opportunity to focus on our capabilities as a supplier. At Obatala Sciences, so much of the work that we do in the lab is to advance products with the experience of the end user in mind. And I think that this kind of forum really highlights that focus. So kudos to the 3Rs Consortium. We welcome the opportunity to partner with you as a provider of human cells and biomaterials, as well as a collaborator to incorporate aspects of human diversity in new and existing microphysiological systems. And just to close, I'll highlight some members of our OBA team, some of whom may be familiar as 3Rs committee members, including Dr. Trivia Frazier, our co-founder and CEO, as well as Dr. Cecilia Sanchez, our Chief Scientific Officer. And I've highlighted a few members of our scientific team. And in general, I'm just so fortunate to work with a wonderful team of scientists and innovative thinkers at Obatala Sciences. So thank you for your time today. And otherwise, I welcome the opportunity for questions in our Q&A session, as well as look forward to hearing about the work of others.
So, hi, my name's Colin Brown. I'm the Chief Scientific Officer at NewCells Biotech. We're a biotechnology firm based in Newcastle in the north of England. We're about 50 strong, and we've been going for about 7 years now. So what we're trying to do is to make really nice MPS models to service the pharma and other industries. So at the moment, we concentrate on 3 models. So we have been really focused on 3 models. So the models that we're interested in are the retina. So a retina is an iPSC-derived retina, and I'll take you through some of the data on that. In addition, it forms an organoid, but in addition, we have a retinal pigment epithelium as well. So I'll take you through that. In the middle of the screen, you've got our kidney model. This is our most mature model. It's complex, and we've been using it for the last 8 years or so. We've worked with most of the large pharma, a very large number of small pharma as well. And again, I'll take you through that. And then finally, on the other side, we have our airways models, where we've got a small airways model, plus we've got a fibrosis model and a cilia beat model. So we've got a few things to try and get through in the next 15 minutes. So if we start with the retina, so we're very interested in making retina. So this is quite a complex model. So it starts by differentiating stem cells, and it takes about 150 days to differentiate the organoid to its fully differentiated state. In this final state, it forms this potato-shaped object. It's about 1.3 millimeters in diameter. We grow them in 96 well plates. And when we get to about 150 days, you can see that it contains all the major cell types that you'd expect, and they are in the correct orientation. So from the outside of the organoid, we have the photoreceptors, and moving in, we have all the other cells. So it forms a structure, it's a structured system. In fact, if you cut them in half and put these onto an electroplate, then if you shine light, then we get a signal, and the signal differs whether the light's red, blue, or another color. So we generated these retinal organoids. They're available either as a product, so you can come to us and we'll sell you retinal organoids, or else we can do in-house service. If you've got a project that requires retinal organoids, then we can discuss how best to approach that. What sort of things have we done with retinal organoids? We've done quite a few things. So in the top left, we've looked at drug safety and efficacy screening. So we've looked at drugs that are toxic in the eye. We've also looked at drugs and trying to deliver drugs to the eye. We've also been in very important part therapy for the eye is gene therapy. So we're looking at gene therapy and vectors and delivering genes to the eyes to correct genetic defects. Another focus that we've got is a developmental study. So many people are interested in how the retina develops, and so that we have our retina at different stages of development, and at different stages we have different cell types are at their peak production, so that we can sell retinal organoids at different points during the developmental cycle, so that you can look at development. And then finally, we have got disease modeling, in which we can not only make retinal organoids from normal tissue, but we can make retinal organoids from diseased tissue. And then that allows us to go back into gene therapy again to treat the disease, or into other methods of drug safety and drug efficacy to look at drugs that may affect the retina. So the other part is that when we make a retina, although we have a substantial amount of RPE cells, it's not fully covered. And so we've made a separate RPE model. The separate RPE model we grow on thin cells, which are transparent transwells, and we're able to look for the functional markers that you'd expect to see there, and we've proven that they're very pure and highly differentiated, form a barrier, and then we can look at marker expression and transepithelial resistance. So again, this gives us a product to look at drugs that may affect the retinal pigmented epithelium, either as a treatment or as toxicity, as an off-target effect. So we have both retinal organoids and the retinal pigmented epithelium. So again, these are available both as a service and as a product. Jumping ship into the kidney, we've had this kidney model for a long time now, and it's been very well used. So what does our kidney look like? Well, we are very lucky in Newcastle in that we have very good access to human tissue. In fact, we get one complete human tissue per week. So we get about 80 million cells per week so that we can do make models. So our model is essentially based on a transwell. I mean, we published this quite a few times now. And on a transwell, the cells form a monolayer, which represents the barrier function of the tubule, plus it expresses all the transporters. It's an MPS model in that it not only has proximal tubule cells, but it also has some distal tubule cells, and also it generates its own ECM. Now, this generation of distal tubule cells is very important because we can look at signaling between the proximal tubule and the distal tubule cells. So we've been looking particularly at the regulation of phosphate transporter by signals from the distal tubule. So it's well polarized, differentiated, forms a functional barrier, and it's available. Another feature is it's available in cross species. So we have human, dog, rat, mouse, and on the rare occasion that they're available, we can make this from NHP. It has a high level of transporter expression. Very important to point out that it expresses oats and megalin and cubulin, so we can look at not only small molecules, but we've got a really long track record now in looking at biologics and other large molecules. Here's just some data that we really like to show people. The model forms a monolayer, as we said before. It has cilia. You can see the cilia, these tiny yellow dots. It's the red pericentrin on top of a green acetylated tubulin background. Lots and lots of tiny yellow dots. So each of the cells has a cilia. It expresses high levels of OAT1 and OAT3 at the protein level, and it also expresses megalin and cubulin at the protein level. Now we can do lots of things with our model. It splits really into two: DMPK and toxicity and drug safety. So this is just an example of how we can use it in DMPK. We can look, for example, at the handling of a molecule like paraminohippurate, which is cleared by the kidney. And what we can show here is that in our human cells, the secretory flux from the blood to the urine is 3 times that from the urine to the blood. Therefore, it's net secreted, preventative sensitive. And we can make our dog model and we see exactly the same. We can make a rat model, see exactly the same, make our NHP model, see exactly the same. And then we've used this to look at cross-species differences. So not all molecules are handled exactly the same in animals, as we know, because when we come first in man, about 50% of molecules haven't been predicted as how they're handled in human versus how they're handled in animals. The other side of, after we've done ADME and DMPK, the other side is drug safety. And to look at drug safety, we've generated a series of a platform which allows us to look at 6 endpoints of toxicity. We've chosen to look at 3 translatable biomarkers that are clinically relevant and approved by the FDA. So we always look at clustering, KIM1 and NGAL. In addition, we look at TIR as a measure of integrity, ATP as a measure of cell health, and LDH as a measure of cell death. And you can see in this picture here, this is a molecule that's safe. It doesn't change any of the markers. Here's a molecule that is very toxic. You can see dose-dependent increases in all the biomarkers, drop-off in integrity, drop-off in ATP, increase in LDH. Now, to validate a model, we went and did a study with Takeda, in which they gave us 36 compounds. We published this in 2020. The take-home message is that we have a sensitivity of 94% and a positive predictive value of 92%. And that's higher than you would see in an in vivo model. So we've got really nice kidney model. So just to summarize, kidney, we can make it from different animal species, including human. We can look at toxicity. We can look at drug uptake, transport, drug interactions, and we can do this very important cross-species evaluation. So that's our kidney model. And then finally, I'll move on to our lung model. So our lung model, we've been focusing on for a little bit of time. Again, this is a primary model. So essentially, where we're looking at is we're developing two models. One is we're trying to make a small airways model, epithelial cell model, and then the second is we're quite interested in fibrosis, because we really want to look at models that allow us to look at these disease states here. So the fibrosis model, essentially, it works incredibly well, and we launched it about 6 months ago. And so what we have is, if you have quiescent fibroblasts, then if you expose them to TGF beta, then you can cause them to activate, and then they transform into myofibroblasts. We can use this model to then look at other stimuli that causes fibrosis to occur, but equally we have data in which we can reverse fibrosis. So that's allowing us to do that. So we use lots of different endpoints. Here we're just looking at smooth muscle actin. Smooth muscle actin goes up as we increase the fibrosis, but we also have it for collagen and other markers. So we have this really nice assay which allows us to look at the development of fibrosis, but also the reversal of fibrosis, just looking at these fibroblasts. The other model we've been making is, and we've heard a couple of people mention this, is an airlift model. So we grow our from a basal cell. We then differentiate this into a fully formed airway, and once it's formed a functional airway, then we raise the airway up, and then we have it in the form of an airlift. It's very polarized and differentiated. It shows functional junctions. It has a tier, and you can see the barrier permeability here. The other thing is, if you look at the function, the ciliated cells, you can see all the different types of cells that we have when they appear within our system. So the basal cells, club cells, goblet cells, and cilia cells. And we just recently developed the model so we can look at beating cilia. We've developed some brand new software which allows us to really have high definition measurements of beating cilia. And this, if you do have data and don't have good software, we are open to collaborating with people to analyze their data on our system. So if we move on one more slide. So again, we've used it in different ways. So we've been looking at effects of drugs, for example, on toxicity, TR, LDH, ATP, but also we've been looking at the effect of drugs on cilia beating frequency. And so we can show some data here where we can look at the efficacy of both slow and fast-acting drugs in generating abnormal physiology, and we can look at the data using our proprietary cilia beat software, which I've just mentioned before. So, with the time of an essence, what we've got is 3 models: our iPSC-derived retina, which is probably the only retina model on the market. It's highly differentiated. It's highly reproducible. And it's available both as a product and as a service. We have our standard kidney model, in which we can look at DMPK or nephrotoxicity. Again, very complex model. We've been using it for lots of years. It's very well validated. And then finally, we have our lung model, which is small airways epithelial cells, and a second human lung fibroblast assay. And then the final part of that is we've developed a new beating cilia model, which it really does recapitulate the cilia beat in vitro. And we have developed a suite of software programs which allows us to analyze that in great depth. So with that, I'd like to just say thanks for listening, and I can answer questions at the end of the session.
So good afternoon, everyone. Thanks for joining us today. It's been really great to have the invitation to speak here today. So thanks so much to the organisers for allowing me to present to you here today. So I'm Victoria Marsh-Durban. I'm Director of Custom Organoid Services at Molecular Devices, and I'm going to be telling you today about some of the work that Molecular Devices has been doing to really enable the use of 3D biology for drug discovery applications. So really, I don't think this slide needs any introduction at all to this audience. I think the problem that we're all here to try and solve is really that drug development takes an incredibly long time. It costs an awful lot of money and has a very low success rate. Obviously, what this means is not just a waste of time and money and resource going into these programs, but it does mean slower development time and fewer drugs to patients, which I think is what we're all here to do. Again, this slide really needs no introduction here, but again, just to say that at Molecular Devices, the way we see the world is really that obviously the main contributing factor to this massive attrition rate within drug discovery is the use of poor models within the drug discovery process. And of course, what we're looking here to develop, similar to everybody else on this call today, is to really enable the use of more relevant human models earlier in the drug discovery process. And what we mean by human relevant models is really, more close to patient type models, obviously in vitro models, and ones that can be used throughout the whole process of drug discovery. So right from the earliest stages of drug discovery all the way through to late stages of drug discovery. Of course, we recognize that at some stages, there's going to be points at which these models are used in combination with animal models still, and for some time into the future potentially. But really what we want to do is drive the use of models throughout the entire drug development process. And of course, when we're talking about human relevant model systems, these vary in complexity. And of course, we've already spoken a lot today about the pitfalls of 2D cell culture and simple models, and even where those simple models are put artificially into 3D culture, such as with spheroids. And I think, where we're really driving to use is these much more complex models. So these include organoids, organ-on-a-chip models, and microtissues. And of course, everyone here is going to be familiar with those, but just to say primarily within my talk today, I'm going to be focusing on the use of patient-derived organoids, although of course we do support other platforms within Molecular Devices. So of course, with using these beautiful, much more representative and much more data-heavy human relevant models, with that complexity comes additional challenge. So we went out to our customers and we asked them what were their main challenges with using 3D cell culture for their particular applications. And this slide really summarizes a lot of data going out and talking to a lot of people. But really what we found is that the top 3 challenges that customers have when using 3D organoid models within their research is firstly, the ability to scale the amount of the model that they need. Secondly, being able to access sufficient models that represent patient diversity, so being able to scale out to different types of models. And thirdly, dealing with batch variability and reproducibility within these complex models. Now, obviously, that's a very fine balance to strike, because really these are models that you want some level of variability within the models. That's what makes them good models, but clearly that needs to be controlled in terms of the variability and reproducibility to be able to make those able to be deployed into end assays. So just to say a little bit about the portfolio within Molecular Devices, I think most people probably know Molecular Devices. They might even have some Molecular Devices instrumentation within their own labs. But really, historically, MolDev has been really known for analytical tools such as microplate readers and high content imaging. And the area that I'm going to talk a little bit more about today is our 3D biology segment, which is rapidly increasing. And within this area, we really have 4 main areas that we're looking to support 3D biology within our customers' labs. So firstly, we have our organoid production technology, which is, again, the area that I'm going to be focusing on today through the rest of the talk. But just to say, to complement that, we also have some really great automation technology. So the Cell Express AI is our recently launched all-in-one cell culture system. We have the Organoid Innovation Center, which is basically customizable automation workflows, which are geared and focused on 3D biology and 3D model generation. And then finally, we also have the Bioassembly Bot 400. So the BAB is a 3D bioprinter, which enables, again, more complex assembly of 3-dimensional models. So to continue with my talk and focus really on our organoid production technology, again, this is a technology that was acquired by Molecular Devices at the end of 2022. It was acquired from a UK-based biotech called Celeste Limited. And Celeste had really developed an innovative bioprocess workflow to scale 3-dimensional patient-derived organoids at scale. It's a very flexible workflow, so we can scale both our in-house lines to make off-the-shelf consumables of organoids, but we can also expand our customer's own line on a fee-for-service basis. The key to our process really is that it's real standardization of the workflow, standardization of the model output to give really the most consistent models that you possibly can at the end of this workflow. Of course, the other great advantage here is that the organoid products that we develop at the end of this process are provided in assay-ready vials. So it's basically bulked up, frozen, assay-ready organoids in a vial. These are stored by the customer in the freezer and can be thawed at will. And obviously, the great thing about this is it's a platform technology. So when you're thawing those organoids, they can then be deployed into standard plate-based format. However, we've also worked with collaborators who have put these organoids into true microphysiological systems, so into organ-on-a-chip type systems. We've worked with customers who have put our organoids into transwell inserts to make barrier forming assays. And so really, it's just giving you the starting material, that biological heartbeat of the future model that it's going to go into. Of course, always provided in an assay-ready format, so very easy to use, and no real skill needed in terms of needing to understand organoid culture to be able to produce this. A question that we commonly get asked is around our bioreactor technology and what that looks like, and whether the technology is likely to impact or in some way impede the model that we're scaling. And the answer to that is really that our bioreactor technology is designed very much in parallel to manual culture processes. So typically, patient-derived organoids are grown in matrigel domes. So we keep the organoids in matrigel, we grow the matrigel, we seed the matrigel as a thin layer. So it's, the organoids are still grown in the matrigel. What they see is that matrigel or that gel matrix environment. The culture media is placed on top of the matrigel, again, very similar to dome culture. However, the difference is that this is a fed plate bioreactor design. So the media is continually perfused on top of the organoids. And basically what that means is that the organoids have a really homogeneous and consistent environment within which to grow. And what that basically gives us is a much more homogeneous and reproducible product that comes out at the end of this. And again, this is completely platform technology. So really, this is compatible with any organoids that will grow in a gel matrix. So any organoids of different tissue types. We've even started working on iPSC-derived organoids in similar formats. So there really is, this really is platform technology for organoid production at scale. I've already mentioned that the organoids that we produce at the end of the process, we call them 3D ready organoids, which means that they're basically frozen and ready to go into assays. And we also provide these in a small product portfolio from a variety of different tissue types. So at the moment, our portfolio is made up primarily of breast and colorectal cancer, but we also have normal duodenum, and we're working to develop further normal intestinal lines for use in GI tox applications. Again, having already said that one of our most common questions is whether our bioreactor technology impacts the biology of the models that we're developing, I just wanted to spend 5 minutes going through a few pieces of data to show that this is something we've investigated. We've developed an application note, which is available on the Molecular Devices website, to show that the organoids that we're growing are not any different to manually grown organoids in culture. So what we've done here is we've taken our standard manual culture of organoids. We've either continued to expand those manually in culture in standard dome culture, followed by harvesting and putting them immediately into assays. So this is much more comparable with the standard workflow that you would do in the lab when growing organoids on a continuous basis. We've compared that to bioreactor-based expansion of the same organoid line, followed by harvesting of that organoid line and freezing down into our 3D ready assay format, followed by the thaw step before going into assays. So really, this is a true head-to-head comparison of what it would be like to take your organoids straight from manual culture versus taking organoids from the freezer, thawing them and plating them to use into assays. And unfortunately, I haven't got time today to go through all of this data in detail, but really just to say that when comparing manually grown organoids to bioreactor grown organoids, what we found is that the product types were very, very comparable. So we looked across marker expression, we looked at somatic mutation analysis through whole exome sequencing, we looked at gene expression analysis through RNA-seq, and we looked at functional drug responses in these lines. And what we found, as I've already said, is that the manually grown organoids versus the bioreactor grown and frozen and thawed organoids showed extremely similar responses. And also you can get a hint from this particular graph that's on the screen right now, which basically tells you that the error bars on these graphs are much smaller with our bioprocess grown organoids, meaning that the product is much more consistent and much more reproducible. So again, really just to say again that what we've compared is manual organoid production versus using 3D ready organoid products. And of course, these products also have a significant time and cost advantage, because rather than needing to grow the organoids manually yourself in the lab and go through rounds of passaging, dissociation, reseeding and expansion before plating into your assay, what we're providing really is a bulk batch of organoids produced, which can be stored in the freezer, and you can just go to the freezer and pick the organoids when you're ready to run your assay. And as I've said, regardless of what format that's in, whether that's in plate-based format, as I'm showing here, or in a different format. So it's really as simple as going to the freezer, pulling out your organoids when you're ready to go, allowing them to recover for 2 days once they're thawed, and then going ahead with your assay. Just to say again, we've done some more recent work to really look at how reproducible our bulk produced organoids are, and to show that they're compatible with high throughput screening approaches. So again, this was all plate-based assays. So what we've done is we've taken the NIH assay guidance for high throughput screening, and we've taken our organoids direct from the freezer, plated them out, and looked at the reproducibility across the plate in terms of Z prime factors, and also looking at the coefficient of variance between treatment wells. And what we found is that we were successfully able to meet all the criteria needed to pass this assay for use in high throughput screening. So this was really great validation that our product is compatible in this stringent environment. And then finally, the bar graphs on the bottom left of this slide are just showing that batch to batch, we see consistency in terms of the number of organoids in the VAR and the size of the organoids in the VAR. And again, this is what gives us the great batch to batch and vial to vial consistency with our products. So again, just to say that the two points that we're addressing in terms of customer concerns with using these models is the difficulty to scale enough product that's suitable for screening. So we've overcome that challenge, and also overcoming the challenge of the variability in cultured materials. So rather than needing to compare different passage numbers or small amounts of material produced manually, we're really giving that bulk product that can be used all in one go, or gone back to on a repeated basis time and time again to conduct the same assay. And again, just to give you a little bit of a flavour for what's in the rest of the molecular devices wheelhouse. We've also used our bioreactor grown organoids for our automated culture workflows. So again, just saying that our organoids are compatible with varying different workflows, whether that's onward manual culture or automated automated seeding into, for example, the CellExpress AI platform. And so then really just to round off my presentation and conclude, we saw at the beginning that obviously the use of more complex models, there's a strong drive for this within the field, and the need to use more human relevant models earlier in drug discovery. However, we've observed a number of different objections from our customers in the space around the ability to use more complex models. And I'm hoping what I've showed you today is that we're starting to overcome some of this complexity by being able to produce organoids at scale and in a 3D ready format. So just to say, the boxes that I've just grayed out are really the ones that we've addressed with the technology that we've developed, and the ones that are left on the screen are really addressed in other places within the molecular devices portfolio around automating and standardizing protocols. And with that, I will conclude, and thank you for your attention. Happy to take questions at the end in the roundtable.
Okay, hello everyone. So I'm Nicole from Alveolix, and for those who are unfamiliar with us, we are a company. Company based in Switzerland, specializing in organ-on-chip technology. And our mission is to model the smallest functional unit of an organ. And with our Alveolix barrier-on-chip system, we aim to mimic the motion of any organ barrier. And our goal is to create an in vivo-like environment. And here on the right side of the picture, you see our system, of our Alveolix barrier-on-chip system, which is composed of 4 key components. Here we have the actuator, which is the bigger instrument, which is necessary for applying 3D stretch motion. Then we have the exchanger for medium exchange or tear measurement. And then we have the connecting platform, which holds the X12 consumable. And with the actuator, we can really change the amplitude and the frequency of the 3D stretch, depending on which organ you want to work with. So the X12 microfluidic chip is engineered in simplicity following a 96-well plate format, and the two 6-well chips on the AX12 plate make up 12 individual units. And each of those units is comprised of one inlet well, one cell well, and one outlet well, connected by microfluidic channels. And the inlet and outlet wells enable medium exchange from the basolateral compartment of the cell well by the action of the exchanger. The cell well, on the other hand, sits in the middle of each chip and has an extremely thin and porous elastic membrane that splits the whole cavity in an apical and in a basolateral compartment. And this porous membrane serves as a support for cell attachment enabling the establishment of monocultures or co-cultures, such as the one we observe here, where we have epithelial cells on the apical side stained in green, or on the basolateral side, the endothelial cells stained in red. And also it's an open system, so it's also very easy to add immune cells if you want to have an immune-competent model. And also because the membrane is very thin, the cells are in very close contact to each other. So as today's webinar focuses on the cell supply, I will discuss the types of cells we work with at Alveolix, and my main focus will be on the distal lung, but I'll also briefly touch on other organs we work with towards the end. So we offer primary human alveolar epithelial cells, immortalized human alveolar epithelial cells, and organoids derived from patient material, and all of which preserve key organ characteristics during expansion and storage. And I would like to emphasize that our primary epithelial cells are highly customizable. So we can provide HLA-matched models, donor-matched models, tailored donor requests. We can have specific donors for deceased models, and we also can provide clinical donor information. Also, the cells are immune-competent compatible. Our immortalized human alveolar epithelial cells are immune-competent compatible as well, and we also offer HLA-matched models with those cells, and the cells also allow for reporter induction. Additionally, our alveolar organoids can be derived from healthy or from tumor tissue with pre-screening options available, and we also offer the same customization options we have for our primary cells. I would also like to quickly mention that we will offer the same options for gut cells, which are set to launch in 2025. So I will start to just talk about the primary human alveolar epithelial cells and show some data about characterizations. So the alveolar epithelium comprises two distinct cell types, which is alveolar type 1 and alveolar type 2 cells. And with the flow cytometer analysis, we wanted to check markers for those specific cells. And here we can see that it was shown that 99% of the cells were positive for EpCAM and epithelial marker, and 95% of the live single cells express 82 type markers. And data represents 10 donors and can also be found in the paper published by Sengupta et al. In 2022. To characterize the cells on the AX12 support, we did some immunofluorescence staining with an SPC marker and another 82 marker, and which you see here on the top left. And we could prove that with the staining that both 82 type markers are expressed on protein level. And this was also confirmed by immunohistochemistry staining, which you see on the top part of the slide. Additionally, we also, with the transemission microscopy, we could show that the microvilli structures and lamellar-like vesicles were seen, which are specific for 82 type cells. Also here on the top, on the bottom of the slide, you can see another immunofluorescence picture where the cells were stained with Z01 in red, and which is a tight junction protein, and SPC in green. So when looking then at the gene expression of 82 type markers, ABCA3, SFTPC, and SFTPP, we see that the primary cells, which are highlighted in gray, are enriched in those 82 type markers when compared to tissue samples, which is highlighted in dark green on the left side. And the AT2-like character is then preserved over time, exhibiting a high fidelity within the time frame analyzed. And the typical AT2 marker SBC declines over time, and still representing a significant subset of AT2-like cells are present, especially when compared to other suppliers, which will be shown in another slide. Then, when looking at AT1 markers at gene expression, we see that an increase over time is seen with caveolin-1, or with other markers just as OpX or aquaporin-1, we see that they stay at the same level. The Alveolix primary alveolar epithelial cells, which are highlighted here in blue, we are also compared to other primary alveolar epithelial cells from other suppliers, which are highlighted in gray on the gene level for the alveolar markers. And the analysis was performed on day 0, day 2, and day 7. And here on top, we see that our cells have a good expression of AT2 markers on gene level for SFTBC and ABCA3. And AT2 markers, which you see on the graphs on the bottom, such as bodoplanine, tend to have a lower expression compared to other suppliers. And caveolin-1 showed a similar expression compared to the other suppliers. So in summary, it can be said that the Alveolix human alveolar epithelial cells express a marked AT2 character, characterized by specific AT2 markers and lamellar body-like structures. That the primary cells have a marked AT2 character superior to other alveolar epithelial cell providers, which are characterized by an AT1-like character. And then the AT2 character of the cells potentially decreases over time, while the AT1 character potentially increases over time. And our partners choose this model due to the high in vivo-like target expression, the possibility to donor and HLA match, and the clinical relevance. But if you are looking more for a cell line, we do also offer immortalized human alveolar epithelial cells. And hereby, those cells are characterized by building a tight barrier, showing a stable tier above 1000 centimeters for up to 3 weeks. In submerged condition, the expression of tight junction proteins, such as the ZO1, is also expressed, which you can see here on the right side in the immunofluorescence picture, where it's ZO1 is stained in green, actin in red, and the nucleus in blue. FACS analysis was also performed to determine the surface expression of those AT1 and AT2 surface markers, and it was seen here as well that 99% of the cells express EpCAM, the epithelial markers, and surface expression of the alveolar type 1 marker HD156 was expressed in over 95% of the single viable cells. Also, the surface expression of mucin 1 was detected, which is a marker to be higher expressed, to be seen to be higher expressed in AT2 cells. And as a negative control, the nerve growth factor receptor CD271 was used, which is normally used or is a typical marker for upper airway cells, and seen to be negative. And these data can also be found in the data published in 2022 by Sengupta. Protein and gene expression of the cells were also determined and on the chip. And here we see that the fluorescence imaging revealed that the cells express epithelial markers such as EpCAM and mucin 1. Also, the AT1 cell-specific markers OpHex and HD156 were expressed, supporting an AT1-like character. And also here on the bottom, you see that the cells also express the AT2 cell-specific surfactant protein SBC and ABCA3. Highlighting the present and coexistence of both alveolar epithelial types on the AX12. On gene level, the expression of epithelial marker was again investigated, and also alveolar markers. And here in green, you see the alveolar immortalized alveolar epithelial cells, and in blue, the A549 cells, which are adenocarcinoma cell line, and it was used as a comparison. And it was shown that the epithelial markers, such as the mucin, acatarine, and also the tight junction protein 1 are expressed on similar levels. Also, the AT1-like marker caveolin-1 has similar expression. And for the 82 type markers, ABCA3 and the SFTPC, it was expressed in both cell lines, but with a much higher expression in the immortalized alveolar epithelial cells. And this data can be found also in the paper from Sengupta, published in 2022. So in summary, we can say that the Alveolix immortalized alveolar epithelial cells express key 81 and 82 markers. They form a tight barrier with stable values above 1000 square centimeters and express tight junction proteins such as the Z01. And the Alveolix immortalized alveolar epithelial cells are a robust and reliable model of the alveolar barrier. So we do offer pre-seeded cells on the AX12 plate, referred to as cells on chip, AX12C. And these ready-to-use cells have already been shipped to numerous customers, allowing experiments to begin immediately without the need for seeding. And this saves scientists time and speeds up the entire experimental process. The package kit includes both the cells and the culture media, ensuring optimal growth and a seamless user experience. And additionally, the cells on chips can be shipped worldwide. So how can we work together? You can purchase the system, which you saw earlier in the first slide. In addition, you can get ready-to-use cells and save time for your experiment. And we also offer CRO services where we work together with you and find the best suitable model for your research. I talked about lung models today, but also we have established a gut model, also available for CRO and cells on chip. Kidney and bladder is under development and will be available soon. And also other organs are in the pipeline and will follow. But in general, the system works for all barrier models. So finally, I would like to highlight the applications of our barrier on chip system. It can be used for safety testing in healthy models where drug candidates with different modalities, such as TCP antibodies, can be tested. Here also we have data published in Nature Communication from 2024 with our primary cells, which you can look up. Then our system is also suitable for inhalation toxicology, as our X12 blade is compatible with the VitroCell cloud. Hereby, you can find inhalation study data published in the paper 2023 from Sengupta. Moreover, our models can also be used in disease models such as acute inflammation or toxicity, infection models in interstitial lung disease model, COPD models, or inflammatory bowel disease models for efficacy testing. And with these models, we can conduct in vitro ADME tests to better understand pharmacokinetic properties of a compound. And furthermore, also toxicology, safety testing, and targeted therapy can be performed to aid in selecting the most appropriate drug candidates. So with this, I thank you all for your attention, and we are also happy to get any questions. Thank you.
Oh, I did have a few Q&A for all the presenters, and I'll go ahead and ask them, a few of them kind of one by one. Actually, I'm going to start with kind of broader questions first. So first of all, I'd love to hear from the presenters what you all think kind of the biggest challenge is in sourcing cells and selecting cells for MPS platforms right now, like one or two top challenges that you guys find, if any. And I'll, go ahead and you can all raise your hands and then I'll just go in order, or I'll just call you. So Ramia, go ahead first.
I think the biggest challenge is to make sure that it's standardized and it can provide you the robust and validated results. And like, how complex does it need to be? I think we have to choose that accordingly. I think that's quite important to look at. Yeah.
Awesome. Thank you. Anybody want to answer next? Yeah, Dr. Barton.
Hi. You can hear me, right?
Yep, we can.
So one of the challenges, I agree with Ramia, and in addition to that, I also would like to add, especially if we would work for so iPSC derived models or primary models, it's always an issue of reproducibility. going also the direction to standardization. And then it also is important to take into account if you need to do complex systems and donor match the systems, and that becomes even further challenging.
Yeah, thank you. Other thoughts or different thoughts? Victoria.
Yeah, so I think the biggest challenge actually is going to be one that is really not new to anybody, which is being able to choose the right model for the question that you're asking. Like, I still think it's as fundamental as that. I think I see a lot of people who are trying to use different models to answer questions that the model is just not going to tell them, or they want to use a specific model, and they're sort of, you know, they don't realize that actually the model's not going to answer the question that they're asking. So actually, I think that is, for me, still the biggest challenge. And I think as models get more and more complex and more exciting, that problem actually gets bigger. So I think it's really important that people continue to focus on the question that they're asking, the model that they're choosing, and whether that's appropriate.
Yeah, thank you, Victoria. Nicholas.
Yeah, we should not forget about the cryopreservation process as well, and how we could optimize cryopreservation for thawing, cryopreservation and thawing at the same time to establish a good quality cell type that we could use, you know, with, you know, based on how we want and when we want. This is quite crucial, especially when it comes with iPSC-derived cells.
Yeah, awesome. Thank you. Carolina?
Yeah, I think in addition, I agree with what all have shared so far. But in addition, is the quality of the cells as well. I think it's really important to start with material that have a good quality, and sometimes you do have to test kind of a few suppliers until you get the cells that are ensure the quality of what you want, and it's extremely time consuming to be doing that. So I agree that cryopreservation is something that needs to be optimized across the board.
Awesome. Colin, go ahead.
Yeah, so just 2 points. I think we need to work towards standardization, which everyone agrees, because one of the points is we've been very good as plastic engineers, we're less good as biologists. And so I think we really need to have a standard set of compounds that we can test systems in, because I think it's very difficult to say how good a system is if the compounds that you've used are completely different.
Yeah, thank you, Colin. Emma.
Yeah, I think definitely tied to everyone else's responses that when working with early passage numbers in particular, we're often driven by we're constrained a little bit if we're deriving samples and cells from tissue sources of a defined kind of volume weight. Those early passages are kind of constrained in number. So what we often see is we have to engage in some kind of customer education about the necessity of expansion, but also keeping in mind the characteristics that were defined for those early passages in terms of quality and, in our case, like markers of stemness. And once you exceed certain passage numbers, that's a different situation entirely. And then similarly, I think we need very defined methods from a supplier standpoint that you're able to convey to the customer what specifically cryopreservation methods are, even methods associated with different characterization assays, how you're measuring that, and then in their hands, how that should correspond, so that there are multiple check-in points, I guess, from supplier and customer perspective to make sure that we're assessing quality reproducibly throughout that process.
Awesome. Thank you, Emma. Did any of the other presenters want to chime in that haven't already?
Yeah, I mean, I agree with all what was said already before. Yeah, the logistics is a problem to have, especially when you have a really complex model. Also, yeah, the inter-lab differences, right? If you have with the medium optimization for each model, especially also when you have several cell types, that's probably a challenge. Yeah, and also keeping the passage-to-passage robustness. So yeah, I think.
Yeah, thank you. Anyone else that I think most people have said their piece, but
I'll just chime in quickly by saying
Thank you,
I also agree with all of you. And I would just add a little bit of color onto what Emma said, in that I think she's absolutely right. For our models in particular, we need a lot of cells to generate the tissues. And when our customers are expanding those cells, they might start with a pristine quality product, but in the end, just through no fault of their own, just maybe not maintaining rigorous, quality standards in that cell expansion can lead to undesirable or less than desirable outcomes in the final product that they generate. So yeah, just ensuring high quality standards in handling the cells and making the products that you're using to put into the tissues that you're engineering.
Yeah. And Colin, I think you've got a follow-up to that.
Just a final point on that is, we do both services and product. And services, we can guarantee quality. Product, we just find that it doesn't matter how much you write in an SOP about how to handle your cells. People don't read SOPs. I mean, I don't know. And so we find that it's a real problem in sort of getting consistency in when we supply cells to a third party, that they are actually doing what they should be doing with the cells as written in the SOP. I think I don't know how we get around that, and I'm sure it's not just it's not just us. It's not just in our field, it's throughout.
Yeah, definitely. Yeah, thanks for that extra piece.
And I think, just to add on that, it's not even necessarily our customers not following protocols. It's just the fact of interpreting those protocols may be different from person to person. So one person's 80% confluency could be quite different from another. So, how you really maintain those strict standards, I think is one area that we need to tighten up for tissue engineering.
Yeah. So kind of the flip side of that general question, and I think maybe there's somewhat knowing the answer of this, but as from your guys' perspectives, what key pursuits or key, first steps would move the field forward in this area of cell sourcing, cell quality, et cetera? I've heard a lot on we need to standardize and cell quality, but I'd love to hear if anybody could expand on ideas or things, like if there are key things or know of key things that they think would be really beneficial for the field in this area. Emma.
I think we could definitely start by looking at what data is reported on C of A's for cells and standardizing that with what assays are performed, what assays are not performed. Viability post-thaw and pre-thaw and cryopreservation methods would definitely be a start in generating that kind of end user data set that can be readily available when making the decision on where you're sourcing your cells. As it's currently done, that document either is behind many different kind of paywalls, however it's structured on a website. And I think that reduces the translatability vendor to vendor. So we're asking customers to do a lot on the back end to generate documents within their own laboratory structure to quantify and track their cells, when I think we can be doing a lot more from a supplier perspective.
Awesome. Thank you, Emma. Carolina.
Yes, I agree with Emma. I think that's a very good short-term solution, but I think what is going to really move forward in the future is to have products that are close to the assay ready for people to use, that they don't have, to go through the protocols, the validation of the cells or media, co-culture, that in a few days they actually are able to run their assays.
Yeah, awesome. Thank you. Baron.
So our approach, so we are supplying just the iPSC line, but we are not providing assay ready cells, but in-house. What we are doing, and also what we are recommending, is that similar to basically Carolina's approach to either we have a big batch of, let's say, one sort of like, if it's primary, we try to secure a bigger batch to be able to go through the, like, then we know the donor, how it behaves. Same for bone marrow cells. Or in this case, for the iPSC, we do a big batch of differentiation. And here comes then of course, the freezing techniques, the whole cryopreservation into account, that it has to be really great to optimize the cryopreservation. Then we can work with this big batch. I don't know, we have hundreds of files that we can go through the experiments, repetitions, and so on. So that's basically how we are approaching the problem.
Yeah, thank you. Other thoughts